We've now got Angela Middleton from Historic England and Diana Davis from the National Museum of the Royal Navy. And they're going to talk about conservation and analysis of paint remains on leather recovered from a Barracuda aircraft wreck. So I think we can say now for something completely different. Thank you very much.
Hello. Okay. Thanks. Right. I'm going to start here. I'm really just going to sort of sorry, I'm just going to sort of set the scene about what this project is and why these leather objects are important to us, and then I'm going to hand over to Angela, because Angela and her wonderful team have actually done all the work on the leather objects. The story sort of starts in 1944. People don't necessarily think of aircraft when you think of the Royal Navy, but the Royal Navy operate a lot of their own aircraft from aircraft carriers and that sort of thing. And the plane that was made in the biggest numbers for the Navy in the Second World War was the Barracuda. In 1944, there was Barracuda taking off from Maydown Airfield in County Londonderry, Northern Ireland. 3 crew on board. It was a routine flight. They were just going to Scotland for some supplies. Unfortunately, they didn't manage to get more than half a mile off the runway, and something went wrong. We still don't know quite what. And they fell back to earth and crashed at Loch in a section of bog, basically, which is obviously very dangerous ground. There was standing water. The crew went into the bog and were killed on impact. For various reasons, some of which are still a little bit obscure, the Navy, who were on the scene very quickly, chose not to recover the crew. Bogs are very dangerous, but they did have to stand on the aircraft, cut the wings off, and jump on it a bit to make it sink. But anyway, for whatever reason, they chose not to recover the crew. But unfortunately, it seems that they told the families that the crew had been recovered and buried. And when this came to light some years later, there was obviously an awful lot of bad feeling about that. And a campaign grew around recovering those crew after the war. It wasn't until 1971 that they actually managed to do that, because it's quite an operation to float a crane basically across a bog and recover something from the bottom where it was wedged in the peat in waterlogged conditions. So because there was this much kind of feeling and effort around it, that wreckage was then gifted to the Fleet Air Arm Museum of the Royal Navy. And there was a stipulation that something had to be done with it that would create some sort of effective memorial for Barracuda crews. So that recovery was more than 50 years ago now, and the project is still moving along. There are no extant examples of complete Barracuda anywhere in the world, despite the fact these were made in the biggest numbers for the Navy. So there are a lot of wrecks, and there were a lot that were broken up after the war, but there is no single example now remaining. So at the Fleet Air Arm Museum, which is part of the National Museum of the Royal Navy, the project has now taken shape into rebuilding a Barracuda aircraft to sort of complete the collection. It's the only one of its time that doesn't exist anywhere. And it will be based on that original wreck, because that was its sort of intended purpose when it was recovered. Now, the real value in that is what we find from it as we disassemble it, because actually there's very little information about Barracudas now. And obviously picking apart wreckage that is 70 years old and has been at the bottom of a peat bog for 20 of those years is tricky. But team were determined. But we really got a big leap forward in 2019 when another Barracuda wreck was discovered quite by accident in shallow water in the Solent, down off Portsmouth. It was in the way of national grid cable laying, so it had to be destroyed, but the museum got involved and we were able to raise that wreck and use it again to examine it archaeologically and to find out more about how these things were built, so that we could use that information in rebuilding an aircraft. Within the wreck, there were some organic objects, including pieces of footwear. And those objects then become important to us because this wreck could not actually be identified from what was left. So there were two main possibilities of which wreck it was. That obviously becomes important to us because the aircraft were made in different factories. And there's no point picking it apart to discover something when you don't know the patterns that are underneath it. So that is the real value in this project. It's the information that's being revealed about an aircraft that we don't fully understand anymore in order to allow us to rebuild one. And you can see from this schematic, they're pretty big, they're quite complex. There are hundreds of thousands of individual components. We have 7 wrecks altogether now to use as source material for this. But as you can imagine, none of them are complete. A lot of that material's in quite poor condition. And the real sort of joy of the project has been in picking through that archaeological material, finding things that sort of connected to the people who built them as well as the people who flew them. There are little details that shouldn't be there. For example, it's a big no-no to mark aircraft pieces in any way on the factory floor. We're finding little drawings and sketches that people have done of their, I'm going to say girlfriends or wives, slightly saucy pictures here and there, and other little things that to sort of remind you that a human being was involved. So Angela and her team have taken on the leather objects from this, which has then given us the chance to find out more about this material, which feeds back into the project and has really escalated where we've got to at the point. So I'll hand over to Angela.
Thank you. We just felt it was important to provide a bit of background to this whole project rather than just go on and to say, here's some leather from an aircraft, which is the sort of most modern material I've ever worked on. And I know nothing about aircraft, by the way. Maybe a little bit about leather. But so we at Historic England became involved in 2019. Initially, we supervised a student who was meant to work on this leather together with me. But we all know what happened around that time, and it wasn't long before the labs closed and we were all locked away. So the student unfortunately never had the chance to work on this leather. However, we still felt committed to assist the National Museum of the Royal Navy in their quest. And the first question of whether we can serve it, that was a really easy one. Yes, that we can do. That's not a problem. But the second one is, can you use any of your fancy equipment and possibly help identify the aircraft? Was a, I don't know, maybe we'll give it a go. And this is where the amazing work of my materials science colleagues, Paynter and Francesca Gherardi, is much more interesting than the conservation work I did. So I will start with that. The Fairey Barracuda aircraft was designed by Fairey Aviation and manufactured by several companies, including Fairey itself, but also Blackburn Aircraft and Bolt & Paul. So the Solent wreck, this is the most recent discovery that Diana mentioned at the end of her introduction, is thought to be one of 2 possible candidates identified by Wessex Archaeology and the Fleet Air Museum researchers who, and it's described as follows in the Wessex Archaeology report. There are 2 aircraft that were lost within 4 months of each other. They are the same make of aircraft and were both lost due to engine failures and takeoff. One is BV 739, a Blackburn-built Fairey Barracuda, and this was delivered on the 15th of July in 1943. And on the 29th of September 1943, so only a couple of months later, it lost power on takeoff and ditched in shallow waters. The second possibility is LS 473, a Fairey-built aircraft that failed to obtain climbing speed on takeoff on the 6th of January 1944 and also crashed, and that was only completed its build in November 1943. So they had quite a reputation for not being very reliable, and as you can see from those figures, their lifespan was really quite short. So those were the 2 possible contenders So what was the evidence that could be used? And that was actually attached to the leather artifacts themselves. The leather was waterlogged and in really good condition. Artifacts included shoe remains, but also seat leather and other straps and coverings from various components inside the aircraft. The shoe leather was really as expected, but the leather from the actual aircraft had remains of what we assumed was paint, glue, fabric, and metal components attached to it. And as you can see in these images here, quite a lot of the pieces had grey, glossy, but also very brittle paint on them. And also metal fittings and components were still attached to them. And it appeared that the paint remains that we found were originally applied to the metal components inside the aircraft and then had transferred onto the leather where they had been in contact. And then in addition to that, some leathers had also remains of a flexible, semi-transparent textile around the edges. All analyses were undertaken on the wet leather before any conservation measures were applied. And to compare the paint analysis results from the unknown wreck from the Solent, Dave Morris from the Fleet Air Arm Museum provided samples from paint schemes of other contemporary aircrafts to determine whether the paint schemes from the different subcontracted manufacturers of these aircraft could be chemically differentiated. And these samples came from Fairey, Blackburn, and Boulton-Paul aircrafts. Francesca undertook FTIR analysis and found that samples from 2 seat leathers are made of latex. So this is the these remains around the edges on the flesh side here, and with some inorganic additives, silicates that were added to the polymer as a filler. Paint analysis from this one artifact confirms the presence of titanium and zinc white pigments in the paler lower layer of the paint coating, so that is possibly the primer. No binder was found in this layer. And then we have a grey paint layer slightly like on top of the primer, which is likely to be a surviving top coat. It is an alkyd paint with silicate-based pigments such as green earth and lithopone, which is a mixture of barium sulfate and zinc sulfide. Oh my God, you see, that's why I didn't go into material sciences. Samples from different components of this leather assemblage were also analyzed using XRF. This is or can be a non-destructive technique, but the results have to be carefully interpreted as they are affected by weathering and alterations of the material post-burial, but also by adhering dirt and corrosion products. The substrate beneath the thin paint layers could also be detected, and in most cases that was aluminium, as was quite expected for the makeup of this aircraft. Using XRF analysis, Sarah analyzed the main chemical makeup of the paint and primer from samples from the unknown Solent wreck and compared that with the samples of the paint provided by Dave from known manufacturers. And the primers used by different manufacturers of Barracuda aircraft were mainly based on barium, zinc, and titanium pigments. These pigments were present in different proportions and with some distinctive additives, following the paints used by each manufacturer to be differentiated in that way. The primer used by Fairey Aviation was dominated by titanium with some barium and only low levels of zinc, and it also contained antimony, which was absent from the other paints. So knowing all this, the Solent wreck aircraft primer matched precisely the primer from the Fairey manufactured aircraft where we had the paint samples from. And the material analysis, together with some archival research, allowed us then to narrow these results down even further. And it was in fact the aircraft LS 473 and Lieutenant Sands, who flew the Barracuda aircraft that sank in the Solent. So with this combined effort, we were able to positively identify the aircraft wreck remains. And after this amazing detective work by my material science colleagues, sort of the conservation efforts really become almost irrelevant and yeah, not so interesting anymore. But anyway, they still had to be conserved, and this is what I did. So I already mentioned there were a lot of metal components attached to the leathers. And I started off with X-raying the relevant pieces. This was, first of all, for documentation purposes, but also to inform any possible cleaning or removal of overlying concretions that were attached to the leather. And the level of cleaning was decided in an in-person meeting by that time already between, you know, us at Historic England, but also Diana and Dave. And we decided that any overlying concretion and deposits that could tell us something about how the leather was used on the aircraft were to remain on the leather. So only any sort of extraneous deposits or concretions that didn't add anything to the understanding or the story of this piece of leather was removed, in fact. And we also decided, obviously, to leave the paint remains and other deposits on the leather, because we thought this would tell us something about their original use or placement in the aircraft. And this becomes especially apparent on these triangular panels, which form a sleeve or a cover around a gear stick. Equally, creases are an indication of repeated use and were left in place as evidence of such use. So after that sort of decision-making process of how far we would go in the cleaning, conservation then could be undertaken in earnest, and anything coming from a marine environment requires desalination. That was carried out using water changes with distilled water. Then impregnation was undertaken with PEG 400 and drying using vacuum freeze drying. So really nothing too adventurous here. During freeze drying, though, leather will shrink a little bit and some pieces experience distortion. One such example was the insole of one of the shoes, and this was gently reshaped by placing it over a semi-permeable membrane in a box filled with water. And over the course of a couple of days, the leather will sort of become supple in this high humidity atmosphere. And this is where you can then start to gently reshape it again. So at this point, it was sandwiched between layers of plastozote and corex and gently straightened out again. The sandwich boards were secured with ties and tapes and the leather placed back into the box. And as the water then evaporates through the semi-permeable membrane, the humidity reduces to ambient and the leather will remain in a more sympathetic shape that is more appropriate for an insole. So as well as analysis and conservation, the leather has also been examined by leather specialist Quita Mould, who has provided detailed descriptions of each piece. And in a combined effort and using analytical as well as archival research, it has been possible to identify the aircraft wreck remains as a Fairey Barracuda LS 473. The leather, together with the aircraft remains, form a tangible reminder of the past and will add to our, or rather to the National Museum's, National Museum of the Royal Navy's program and involve, an audience, a new audience to this really quite moving story that is not already engaged and interested in the more technical aspects of the Barracuda aircraft that we quite often find. So as become clear, this project has been a real team effort, and I want to express my sincere thanks to our colleagues who generously allowed us to use and present the results of their work here, but could not be here today. And before I finish, I just wanted to remind everyone please take away a little postcard, which I put out at the front desk there. There's a QR code at the back that takes you straight to some more leather work that we have undertaken in the past. Okay, thank you.
Okay, so next we've got Lou Allington-Jones and Ranbir Bailey, who are going to talk about detecting damage to DNA. Do no harm with none of this. And as a conservator, or retired conservator, we always do our best, but we always know that in the future somebody's going to say, you didn't use that. So don't be too hard on us.
I'm a conservative too, so I'm not. Yeah. I think our research has worried us enough that we're actually going to rerun it all again because we don't actually like the results. But yeah, so here we go. So this project actually has an origin story in another project that we started a few years ago, where you've got a problem here. You can see in this image where you've got this skin with some feathers attached, and this skin has turned orange and become very friable and crumbly. And this is a problem in taxidermy called fat burn. And this is actually the lipid oxidation which occurs in the skin, especially if it hasn't been prepared properly. So you end up getting a lot of, so when you prepare a bird skin for taxidermy, you're meant to scrape out all the fat. And this is actually particularly problematic, especially with water birds, and you quite often leave areas behind, especially in the rear of the bird, and you end up getting their bottoms falling off, which isn't great for displays at Natural History Museum. So we wanted to find a solution for this, but we really didn't know what to use. And I was really interested in looking at red rot solutions for the leather conservation community, because fat burn is actually exacerbated by the presence of sulfur from sulfur dioxide. So I thought it might be a similar problem. But we didn't know the effect it would have on the skin because it's untanned and might not be suitable. Things that are okay for leather might not be great. So we wanted to look at pH, shrinkage temperature, and also the effect on DNA, which is really important in natural history collections. So I chose a few treatments from leather conservation and also a couple of things that we use in natural history conservation, and looked at how they might affect the pH. So the red shank is actually historic skin that hadn't been affected by fat burn, and you can see here the pH is 6, so it's not too bad. The control has actually been affected by fat burn and has gone really low to pH 4, and then the treatments we gave it, Cluso had no effect on the control skin, and a couple of the others actually did increase the pH, which is good. I mean, not vastly, but it was going somewhere. I also looked at shrinkage temperature. You can see the red shank shrinkage temperature is obviously lower than leather, tanned leather, Cara leather, but it's not too bad. But the pH, sorry, the TS of the control skin was so low that I actually had to chill the water before running the test, because I sat looking at it for ages and it didn't shrink. So anyone that doesn't know, shrinkage temperature is when fibers start simultaneously shrinking. So you immerse the sample in water and then gradually heat it up. But I actually had to immerse it in cold water and then allow it to reach room temperature. And then I had mixed success. Some of the leather conservation treatments like oxazolidine and SC6000 worked pretty well. So that was really encouraging. So then we wanted to look at DNA because I had no idea about what these treatments, these chemicals might do. And that's where Ranbir came in, because she's the super scientist. And we found that we have a slight damage from a few things, but quite severe damage from things like paraloid B72 and acetone, which is something that we had been using on skin. So this kind of freaked us out. So if you want to look at, just explain what these mean, these are tape station DNA results. If you have a good result, you have a nice dark band at the top of the tape. And if you have a poor result, you get a lot of fragmentation throughout the area, and then when everything's disappearing, that's just appalling, basically. So if you're interested in this project, you can read more about it. I am aware that Stella said earlier that you can't extract much DNA from tanned leather, but hopefully there is going to be a crossover for people who might be interested in the proteins that Stella and Lucy were looking at. So, but in natural history collections, it's really the way that all the research is going. And you can look at things like migration, not only identifying what animal it is, but if you can look even closer where the animal might have lived, where the migration occurred, if there's any trade. And obviously from a natural history side, you're looking at environmental change, migration of species and evolution. So it's really quite important. So we decided to look at a lot more conservation chemicals because we were frightened with parallel B72 is really causing a problem. What else are we doing that might cause damage? And I think this project really illustrates how we kind of sometimes just go along where we've tested these adhesives and we know that they've got good aging properties and they don't chemically interact negatively with the specimen. They don't discolor with age, that sort of thing. And they don't go off gas any nasty VOCs or anything like that. But then new types of analysis emerge. And we need to, every time that happens, we need to look at what chemicals we're adding to specimens again or objects. So we treated a load of skin samples. These were bird skin samples with a range of a load of different solvents, a couple of detergents, adhesives and consolidants. So the solvents, we flooded the sample and drew it off immediately. So that was kind of representing a quick clean. And then these were dried for 9 months because we wanted to look at the longevity of treatment. Any detergents or ammonium hydroxide were rinsed before drying. Consolidants again were saturated the sample and then excess drawn off, but the consolidants and adhesives weren't actually removed from the specimen, they remained on the specimen. And I also did heat to see whether that contributed to deterioration if beaver was used. And then also ammonia vapor, which is something that we recommended for use when we did the fat burn treatment. And we didn't test anything that might actually add DNA, like arrowroot or racing glass or anything like that, or wheat starch. Okay.
So I'm Rambir, and I'm just going to, this is just a quick introduction of who we are at the Natural History Museum. We're the Conservation Centre and Molecular are under a bigger department called, now called Science Innovation Platforms, used to be called Core Research Laboratories. We're headed, SIPS, as we're known, is headed by Alex Sosick, who's our new head of department, but he's acting head of division for molecular at the moment. So I just wanted to give you a quick overview of what we do. And of course, Lou has already pointed out that a lot of our work really overlaps with the Natural History Museum collections, especially of those collections in the archives and all the historic stuff, because we're interested in getting molecular data out of that. So the team, we've got a number of different facets to the lab. Just to give you an overview: facility operations, that's me, that I run the laboratories, you know, the research training for the students, the inductions for the new staff, the equipment, the budgets, it's just anything that goes with running the facility. We've got an R&D arm, including robotics, because we're really trying to, especially if we want to eventually look at all of our historic DNA collections, of which we have thousands and hundreds of thousands, we want to sort of really high throughput. So we've got a robotics arm under our research and development, headed by Piotr and Yun, and then we've got our DNA sequencing team there, and also DNA sequencing generates lots and lots of DNA data that we have to have analysed and looked at by our bioinformaticians, Sylvia and Thomas. So that's who we are, and we all kind of overlap. We don't just stay in our little boxes. We're all encouraged to overlap, and hence I'm doing a little bit of research with Lou, which has encouraged me back into the lab. And so this slide just shows you the diverse range of samples that we work on, and we really are a facility that facilitates the research that goes on in the museum within with our internal researchers on the plethora of samples that you see there. But we all, and our expertise is specimen, collecting that specimen, extracting the DNA from that specimen, amplifying it up, DNA sequencing it. And for all these different kinds of projects. So we do lots of technical and method development, we do lots of species conservation and lots of biodiversity projects. There's a whole range there, and really we also work with lots of external stakeholders as well. So this is some of our activities. This is some of the research, some of the core research team. We all run the facility in our own way, whether that's teaching. We teach a master's molecular practical course, Imperial. We do lots of infill genomics. Our dream is to be able to do everything in the field, from specimen collection all the way through to DNA sequencing, and we've definitely demonstrated that. That's in the gardens at the museum. We do lots of sort of outreach projects, Grow to Know, lots of biodiversity projects. This is Grow to Know is part of Urban Nature Project, and where we've made a prairie garden. Well, they have made a prairie garden at the site of the Grenfell disaster, where they're getting the community involved in biodiversity. So we're taking soil samples and over time and seeing whether we're getting more biodiverse by adding lots of different plants and hopefully attracting lots of different insects. And we've got the Darwin Tree of Life project that's going through the lab at the moment, and of course trying to identify over 60,000 species in the UK Isles. And we, more close to conservation, conservators' hearts. We're doing, we've done, my colleague Piotr has headed an IPM air surveillance project at the museum, where he's collecting air samples from various collection spaces to check what bugs might be, putting our collections at risk. So whether that's molds or bacteria or other pests. So that's the kind of work that we get involved in. So it's quite varied, actually. And so Lou gave me her treated skins. These skins were actually much nicer to work with, because I guess they're fresh and they were much thinner, rather than because the previous the plot that she showed, we had worked on some modern skin and some ancient skin, but this was kind of starting from scratch, a really clean experiment. And so the crux of getting of this experiment was to get good DNA out, so that we could see how disintegrated it was with the various treatments. So 3 basic steps of DNA extraction: disrupting the cell structure so that we can get the DNA out and create a lysate. We then want to get rid of all the contaminants and the impurities and separate those from the soluble DNA, and then we want to recover the pure DNA. And just a very simple slide to show how I did that. The original paper, I was using a grinding method, which we often do for tough tissue. We use sort of molecular grade sand and pestle and mortar, sometimes liquid nitrogen, but I didn't use that this time. And you really grind the tissue up. But that in itself can be quite damaging to the tissue. So this time I did do a test run. I don't think I told you, Lou, with 2 or 3 samples, where I tried the grinding, and actually it wasn't giving me much yield. So then with advice from my colleague Elena, who has used to working with a range of different samples at the museum, she said try bead beating. So that involves just cutting up a little bit of tissue, popping it in an Eppendorf tube with some ceramic beads, and then beating it really, really fast, high speeds for literally seconds, until you can visibly see that the tissue is nice and broken up. and because you're not, yeah, I'm just wondering whether that's why I had much better yields of DNA with all the 33 samples that we provided. And then what we do, once we've got that broken up tissue, we incubate overnight with a lysis buffer and proteinase K, which breaks down proteins, and the lysis buffer does a combination of that as well. It does a combination of protecting the DNA, breaking down the cell membrane, and all of its contents, and also trying to keep away block DNases that might affect the quality, that might break down DNA. And then this time, I used a very used a slightly different method of extraction, where it's a kit, super easy to use. Oh, it's not showing up properly. I don't know what's happened. Oh, there's nothing I can do about that. So the and it's based on a silica resin within a tube that sort of, and it's based on high ionic strength and K-tropic salts that were introduced so that the DNA of interest gets broken down, the hydrogen bonds go, and then it has this affinity for the silica in the filter within the tube. So the DNA of interest binds to the silica on the outside of the tube, while all the other contaminants are sort of washed away, and that they can then be passed through the column. So I just want to make sure I get that right for you guys. So once we've introduced that sort of high salt content, the DNA is safe, and despite all the washes, it stays there until then you introduce a low salt buffer, after which the DNA loses its affinity for that silicon, comes back into the tube, and then you elute it after lots of washing, and hopefully you have your pure DNA. And this is whole DNA, genomic DNA. And so then we wanted to quantify it. This was the interesting bit. So all of the processes you do up to the DNA extraction, and once you get your, they all sort of have some sort of effect on the DNA. I wanted to compare, just as we did originally, 3 different QC methods for the DNA. We've got spectrophotometric method, fluorometric, and a very traditional but automated gel electrophoresis method. The spectrophotometric is based on absorbance. So at 260 nanometers, if light is absorbed, we see the presence of DNA, and it will give you a rough reading of your concentration of DNA. The fluorometric method, it's not overly accurate, but it gives you an idea of whether your extractions worked. The fluorometric method is a way of detecting, based on fluorescence, a way of detecting double-stranded DNA. So the dyes that we use don't fluoresce very much until they bind to their product of interest. So as soon as they bind to double-stranded DNA, because that's what we want, they then really in fluorescent sort of, they intensely fluoresce, and then we get a reading, and that's actually very accurate for us because a lot of our downstream applications involve DNA sequencing. So it's really important to us that that we've got double-stranded DNA, and it's absolutely of the best quality. And then the tape station really is more for looking at the DNA integrity, and it's an electrophoresis sort of automated system that cuts out all of the traditional sort of agarose gel and all that handling that you have to do, and it's brilliant. It takes 20 minutes, and you load your samples, and it gives you lovely traces. And so I'm going to show you some traces later, but this is one of Lou's lovely graphs where she put the DNA concentration of the qubit. This is the fluorescent QC method, just so we could get a rough idea of what was happening. We've got the green line roughly that we put on the control, and then the red line where we kind of thought below that, sort of, roughly half of the control, we could see that whether the treatments were, how they were affecting the concentration of our DNA. And you can immediately see that there's a handful that were really reducing the amount of DNA after treatment. And there were a number of treatments that actually didn't, which was good. This is a gel image of the genomic DNA samples that we ran after all the treatments. And as Lou said originally, what we're looking at, unfortunately, my control doesn't look as nice and tight as I wanted it to, but what we want to see when there's no degradation is a nice high band here. This is lots of long fragments, this is lots of short fragments, and this is the DNA ladder of known concentration, so we can quantify. So if we've got a nice tight band at the top, which I had in the previous plot actually, but here I don't quite know why I've got so much, a little bit of degradation in my control there. It could be all sorts of reasons. And then with the treatments, we can see, as Lou very accurately explained, you know, if we've got a smear here, this is representing lots of broken up bits of DNA. And if we've got nothing there, the DNA is pretty disintegrated. And if we've got lots of heavy sort of band at the bottom, that's lots of short fragments of DNA. So we can immediately see, for instance, that the really severe effects, for instance, pH 5, pH 7, and also the ECOS, sorry, I tried to learn the full names. And just to name a few, and then of course you can see immediately sort of the reason, the ones that gave us not very much of a destructive effect on DNA, such as the BVA371 and the ethyl acetate, and the Stoddard, for instance, and LASK303HV, which I don't again know the full name of, even though I have it written down. So really, that's what we wanted to see, a range, and I compared, you know, so we've got a number of really good treatments, a number that show that there is quite a severe effect on DNA integrity, and a number that actually, if you can see here, a number of them, such as paraloid B44, paraloid B67, and methyl C, you know, the DNA concentration still remains high, and actually hasn't been affected yet. The integrity of the DNA has. So it just means we've got lots of DNA there, but it's just very short fragments. So it has had an effect on breaking down that DNA. Unfortunately, but I mean, to be honest, if you've got DNA, you know, if someone wanted to go on and sequence this, you know, there are different sequencing techniques that you can use to get a decent result from that. And these were our sort of overall conclusions. I don't know what the S, oh, the S is different, sorry. So there you go, there's our good, bad, and well, good, moderate, and severe, actually. And then, as I explained, there's others that have not affected the DNA concentration, but have caused quite a high fragmentation. And we hope to repeat this work, just so we've got our results absolutely right, and then look at the effect of treatments on bone and the DNA. Thank you very much. Oh,